Great job working through the previous two tasks on how data is translated from field sheets to a file, and you're now familiar with metadata. In this final task, we'll use our assigned sites and months to calculate population size using NEON's small mammal trap data. Make sure that you know what plots and months you'll be analyzing because everyone has a unique combination of plots and months that we'll use to calculate population size. So the first month will serve as the first sampling event to calculate N1, and the second month will serve as the second sampling event to calculate N2 and M2 using our Lincoln-Peterson equation. Then, when we have the values for these three variables, we can estimate N, or population size, for your assigned time and plot. There are several students who have the same plot ID and time frame, and these will serve as our validation to make sure that we're getting the same answers. Then, once everyone has posted their calculations in the discussion forum, we can brainstorm how and why population size changes over time. So let's navigate to our data sheet. This is the raw mammal per trap night data. Right, you can see it has this very long NEON-specific file naming structure. It has the organization, the domain, the site ID, and the data product ID, which in this case is the mammal data. And notice that this is a .csv file. With this type of file, any special formatting or if you create additional tabs, those will not be saved when you save the file. Those will not be retained when you save the file. And this is the major difference between .csv and the normal Excel file format, the .xls. And that's important to understand. In a few weeks, we'll learn how to do everything that we're going to do today in R, but it's important for you to understand how this is done in Excel so you can compare the two approaches to really understand why R makes analyses reproducible. Today, we'll be doing a lot of point and clicking, and that's difficult to replicate in an automated way. So you've got the file open. You should recognize the column headers as the variables listed in the NEON metadata files. You should also recognize the tidy data structure. So a single column has a single type of data. Each row is a single observation. In this example, in this data, it's an observation for a single trap. You should also recognize that a single cell has just one type of data, as well as in the other tidy data rules that we talked about a few weeks ago. The data we're working with is from the Smithsonian Conservation Biology Institute, a few hours drive from Richmond. And the first thing we want to do is to isolate our specific plot and months of interest. To do this, we will use the filter function in Excel by navigating to the data menu and selecting the filter icon. I'm going to select, so data and filter. When you select the filter icon, you should immediately see these drop-down menus under each column header. So let's navigate to this plot ID column, select the menu arrow, and you should see these options pop up. We want to unselect the select all, and you want to select your plot ID. In this example, I'm going to work with plot ID SCBI_002. We can see when I press OK, we have a new set of data, and we can see here in the bottom that there were 1,802 records of the total that met this criteria of being plot ID 002. I can also see that there are a lot of rows where the trap was set, but it was empty. And I don't need this data. I want to select the rows where the white-footed mouse was actually caught. So I'm going to scroll across and find the column called scientific name. And I want to unselect all option and select the species that we've been working with, this Paromyscus leucopus. And I'll select OK. And again, I can see that filtered even more records. So now I'm down to 89 records that met those two criteria from plot 002 and that were this species of interest. We need one more filter for the months that we're interested in. So I am going to look for a column called collected date. You might have to expand this to see it all. And under that drop-down menu, I'm going to unselect the select all option. You can expand the options for each year to find your set of months that you're interested in. In this example, I'm going to select September and October because that's the time period we're working in right now. And so I'm kind of curious what the mice population is up to during this month, way back in 2014. After I selected that filter, you can now see we're down to 14 records or rows that met all those filter criteria. Plot 002 for this particular species, collected in September and October. All right, so my first month of interest is September. And because I'm working with 14, you'll be working with maybe 10s, maybe 20s rows. So it's easy to do this by hand, but you can imagine if you're working with hundreds of rows, this isn't going to be easy to do by hand, but this is an important comparison, how we do it in Excel and how we do it in R. So I want to calculate the number of individuals that were caught during the first sampling event or sampling bout, and that is all the days in September. And when I highlight the rows of interest, you can see that Excel provides you with some summary metrics, including count, sum, and average. In this case, our column is a date, so sum and average aren't biologically relevant, but count is. So I'm going to write that there were 10 individuals in the first sampling event. That is our N1. So write that value down on a scrap piece of paper. My second month is October. So I need to count all the individuals trapped in that month. It doesn't matter the different days. We're counting the month as a single sampling event. And in this example, I count 1, 2, 3, 4 individuals, and that's our N2. Now I need M2, the number of individuals that were collected in the second month that had been previously marked in the prior months. I'm going to scroll down, scroll across to the various columns until I find one that's labeled recapture. So these are the individuals that were marked, that were recaptured from the prior month census. And I can see that of these 4, there were 1, 2, 3 individuals. So M2 is 3. So we can now plug these 3 values into our Lincoln-Peterson equation to estimate population size during September and October. So let's navigate back to our Lincoln-Peterson equation. All right, we have the values of N1, N2, and M2, and we can plug these values into our equation. So I'm going to do that. Right, so we have 10 individuals in the first month, 4 individuals in the second month, and of these 4, 3 were recaptured. If you remember from basic algebra, we can use these ratios to cross multiply. So 10 times 3 is equivalent to 4 times n. In this way, we can start to isolate the variable of interest, which is n, our population size. Again, we can isolate n by dividing each side by 4. So 4 divided by 4 is 1, and then this divided by 4, right? So you see that invisible 1 there. And when we do the math, n is equivalent to 7.5 individuals for September and October for this particular plot. So now you know how to calculate population size from a really large data set. It's your turn now to replicate these steps for your assigned plot and months. And once everyone has posted, we want to see if there's an interesting pattern of how population size changes over time. And this will help us brainstorm the kinds of questions that we can ask using the NEON data that we are interested in.